Good day, everyone. Welcome to our new chapter. This is going to be chapter 6, which will cover the sensory system. All right. Now, our main goal in this chapter is to explore the different types of receptors that we come across when we talk about our complex sensory system. And I call it complex because it can range anywhere from a single cell to a handful of cells, the glands to very specialized organs that are able to receive stimuli and communicate it directly with the nervous system. And the way it's going to work for our class is that chapter 6 will take a look at the sensory system, and then chapter 7 will take a look at the nervous system, and we'll see how one is able to communicate with the other. But before we get into all that, let's talk about the fact that you have different types of receptors based on the stimulus that they're able to react to. And the 6 most common ones will be chemoreceptors, mechanoreceptors, photoreceptors, electroreceptors, magnetoreceptors, and last but not least, thermoreceptors. And as we go through the PowerPoints, we're going to discuss each of these 6 receptors, and we'll talk about how they compare across different animal models. And one thing that you're going to notice from them is regardless of what the stimulus is, when the stimulus meets its proper receptor, you will always have the same reaction, and that is going to be a change in the membrane potential of the sensory cell. All right, so what we're going to see happening, and you can see it on your PowerPoint, are 4 steps. The first one is that the proper receptor will bind to the protein so that you can detect the stimulus. So we'll have the activation of the receptor. And the way the receptor is going to respond to the stimulus is by going ahead and either opening or closing ion channels. And the movement of the ions will cause a change in the membrane potential. Very similar to when we were talking about depolarization, repolarization, action potential. And that change in membrane potential, that excitation, will go ahead and create a signal that is communicated with the integrating center. And the integrating center for us is going to be the nervous system, more specifically the central nervous system, where they'll be able to receive the signal and interpret it and figure out what the course of action will need to be. So here it is from an illustrative example as to the response to the receptor cell. So you can see we are looking at 3 different types of receptors. We have our chemoreceptor, mechanoreceptor, and the photoreceptor. And what you're going to notice is that all of them will have a receptor protein. All of them will have a specific stimulus that they are responding to. But notice on the inside of the cell, they will all respond the same. Once you have the proper stimulus connected to the receptor protein, it's going to go ahead and transduce or convert the signal, and we're going to activate a transduction pathway, and that will cause the ion channels in the receptor cells to respond. Now in this case, all the ion channels are going to open, but there are cases where they will close. But either way, the changing in the ion channels will cause a change in membrane potential. And that change, that signal, will be picked up by the integrating center. So that will be the nervous system, which will be chapter 7, where we're going to take a look at what exactly do I do with that signal, how do I interpret it, and how do I respond to it. So let me give you some examples of the stimuli that will go ahead and correspond to the different categories. And obviously this is just a little quick screenshot because we're going to talk about it in more detail for each of the categories. So if you hear me mention a chemoreceptor, that means that it's going to be a sensory cell that can go ahead and respond to different chemicals. So things like changes in pH, changes in oxygen concentration. And usually when you talk about your ability to smell or taste, that's the category where your chemoreceptors will work best. If you see a mechanoreceptor, that one's going to be perfect for detecting any type of pressure changes or movement. So obviously touch comes into mind, but also things like hearing, balance, heartbeats, blood pressure, and proprioceptors. Proprioceptors are the way for your nervous system to kind of orient or figure out exactly where your body is located in space. So it's kind of like a sense of body position. An example of that would be, for instance, if you go ahead and you hit your elbow, you automatically know that the elbow that you hit is the right elbow, and your hand will go and reach for it to kind of suit it. Or if you feel like you have a little insect bite, if someone says, well, where's the insect bite? Your nervous system is able to go ahead and explain that the insect bite might be located on your pinky finger or on your left hand. So proprioceptors allow us to kind of figure out exactly where our body is oriented in space. Photoreceptors, as you see from the name, the photo will means that it's going to respond to light stimulus. So vision would fall in this category. And then the last three are your electroreceptors, which will respond to electrical fields, magnetic receptors, which will respond to a magnetic field, and thermoreceptors, which, as the name also indicates, will allow us to interpret temperature changes that we become exposed to. Now, before we start exploring each of these receptors and comparing them in different animal models, I do want to go over some concepts that will apply to all of them. And one of them is going to be what we call a receptive field or population coding. So let's go ahead and take a few minutes, talk about that, and then we'll change back to our chemoreceptors and we'll work our way through the list of all 6 of these particular receptors. Alrighty, so here we have an illustration of some sensory neurons. These are some afferent neurons, meaning that they're able to interpret changes to the sensory system. And what we see happening is that they usually will be found in what we call a receptive field. Now, a receptive field is going to be an area in the body, like for instance, an area of the skin that will be able to respond to particular sensing by these afferent neurons. Now, the size of the receptive field will differ. What we see happening is that sometimes you can have a large receptive field and sometimes you'll have a small receptive field. Usually what you have with a large receptive field is that you're able to detect more stimuli because you're covering a larger field that the neurons can respond to, but you can't really detect it in a precise mechanism. So you can't really see small changes in the stimuli. You can basically just detect the beginning and the end, so the threshold and the saturation point. If you have a small receptive field, then usually what we see is that you have neurons that are closely associated with each other, so that once again you can respond to stimulation, but you can also be acutely aware of any slight changes in the stimulus. So it allows you to have more of a precise, specialized analysis of the response to the sensory system. And as we do our different receptors, I'll definitely give you different examples of a large versus a small receptive field. We also see that a lot of times, because you can't be as specific all over the place with the sensory interpretation, is that neurons will work together and create this thing called population coding, which is that they'll tend to overlap the same receptive field so that they can share their data. They also do this thing called lateral inhibition, and that's a really good way to kind of indicate where a strong stimulus is coming from. So for instance, go ahead and take a look at the illustration that's on PowerPoint, and what we see happening is that we have 3 efferent neurons, and we're going to assume that they're embedded in the skin and they're all able to receive a stimulus. Now, what you're going to notice is that you have a weak stimulus on the side by A and C, and you have a strong stimulus by B. This would be, for instance, an example if someone were to pinch you, right? When someone pinches you, you have a strong stimulus that's occurring in that particular area of the skin. Whereas another example would be if someone caresses your forehand. So if you take your hand and you run it down your arm, what you're doing is you're creating an equal stimulus across the entire receptive field. When you decide to pinch someone in this particular location, you're generating a strong stimulus. And what we usually see happening is wherever the strong stimulus is coming from, those neurons will start responding to the stimulus, and they'll start their opening of the ion channels, the changing of the membrane potential, they'll start their communication to the integrating center. But along the way, what they'll also do is that they will inhibit the neurons that are on the lateral side or on either sides of them, and they will diminish the response from those neurons. And by diminishing the response from A and C, it makes it easier for the brain to interpret that the strong stimulus actually corresponds to the location of the B neuron. So it allows you to kind of enhance where the stimulus is coming from. So lateral inhibition is perfect when you have a strong signal surrounded by weaker stimulus, and the strong signal will be able to be interpreted precisely because we're going to inhibit the lateral or the side neurons from continuing on and sending their signal to the integrating center. And you can even see it on the graph that's on the side. Even though all three neurons received some type of stimulus, because B was able to induce the lateral inhibition, at the end of the day, that was the only cell that was successfully able to communicate with the integrating center of the nervous system. All receptors will also have what we call a dynamic range. The dynamic range will allow us to see what the difference is between the threshold of detection and the saturation point. Now go ahead and take a look at your graph, and what it's showing you is that it's comparing the intensity of the stimulus compared to the magnitude of response. And as you can see, at the lowest point you have your threshold intensity, and at your highest peak you're going to have your saturation. And the difference between those is your dynamic range. And if you come on down to the text, you're going to notice that if we're doing our threshold, we are indeed looking for the weakest stimulus. However, we're looking for the weakest stimulus that produces a response in a receptor 50% of the time. So the receptor doesn't always have to respond. It just has to respond at least half of the time. So this will be the minimum amount of stimulus that will give you a response 50% of the time. As you increase your stimulus, what we see happening is that the magnitude of response will correspond and it will increase as well. So you can see that in the graph, the response starts increasing as the intensity increases as well, and eventually we'll get to the part where all the receptors are saturated, they are full, and at that point we have reached our receptor saturated, we have reached our end of our dynamic range, and it doesn't make sense to increase the intensity of the stimulus because there are no more receptors to respond to it. So this right here, ladies and gents, is your dynamic range. Another thing that we can talk about, and I slightly mentioned this to you before when I was chit-chatting about the receptive field, is that it turns out that we can have a large versus a narrow dynamic range. And this is basically going to take a look at if you are able to discriminate between large changes in stimuli or small changes in stimuli. What we see happening is if you have a large dynamic range, I want you to sort of think of that as having a large receptive field. You'll have neurons that will be covering the field, but usually what we see happening is that the neurons are overly stretched out in the area that they have to cover, so they don't have a problem detecting the threshold, and they don't have a problem detecting the saturation point, but they really have a hard point discriminating between small, minute changes in increases or decreases in intensity. So you can detect the beginning and you can detect the end, and you can cover a large receptive field, but you have a poor sensory discrimination, making it very hard to figure out where exactly you have minute changes or where exactly you will have a strong stimuli followed by a weak stimuli. On the other hand, if you want to have more of a sensitive response, then the best thing to do is to come up with a narrow dynamic range. And a narrow dynamic change usually indicates that you have a smaller receptor field. You have plenty of neurons to cover it. In fact, some of them will even overlap so they can do their population coding. And because you have access to so many neurons, it becomes extremely easy for you not only to detect the threshold and the saturation point, but you're also able to detect any minute changes that might occur in the intensity of the stimulus. Another thing that we should discuss is definitely how long does the stimulus remain? How long does the receptor keep responding to the stimulus? And it turns out we can have two options. We can have a phasic response or we can have a tonic response. A phasic response is usually what you see when you have an action potential that is generated either at the beginning or at the end of the stimulus. So not for the entire length of the stimulus, just at the beginning or at the end. This is, for instance, what you experience if you were to, for instance, go into a jacuzzi, right? So let's say you went into a jacuzzi, or if you don't want to go in a jacuzzi, that's fine. Let's say you dry yourself a nice hot bath. When your skin touches the hot water, you're going to notice the temperature change, but before you know it, you get acclimated to it and you don't really notice it as much. In fact, you won't really notice a temperature change until it's time to get out of the jacuzzi or the hot bath, and you notice how the cold air affects the skin. So your body was able to respond to the beginning or the end of the stimulus, and this would then be a phasic step. Another thing, for instance, would be odor. Your sense of smell is very phasic. So for instance, for me, whenever I go to see my parents, my mom loves to cook. So if I go around for dinner time, the minute I open the front door, I have a wave of odors that are coming my way, and I can just smell her cooking deliciousness. But before I know it, that smell has gone away, and you just adjust to it. And if you don't like my example of my mom's cooking, well, shame on you. How about if you go and you decide you want to get some new perfume, or you want to go ahead and get some new cologne? You're going to notice that you'll have lots of samples when you go into the store, and oftentimes they'll also have a little container of coffee beans. The coffee beans are meant to kind of reset your smell so that as you smell the different fragrances, you can have kind of like a repeat or a reset button so you can reactivate that phasic response. Is it possible for a receptor to continuously produce action potentials as long as the stimulus is there? Yes, and that would be a tonic response. So this is, for instance, what we would see in photoreceptors. Your ability to have vision and to have sight is due to the fact that the photoreceptors are continuously being stimulated by the photons that they're exposed to. It's the same thing with a lot of pain receptors. Your body will feel the pain from beginning to end. We do see that with a tonic response, sometimes you get receptor adaptation, which means that if the stimulus stays consistent, the receptor will start to adjust to it, and we do see that the intensity will slightly start to decrease. So if you want to have the same level of action potential or the same level of response, oftentimes with a tonic receptor, you will need to slightly start increasing your stimulus intensity. So here is the tonic versus the phasic receptors in graph form. So let's go ahead and take a look at A. These are going to be our tonic receptors. So remember, the tonic receptor is the ones that will continuously have a response. And what you can see over here is you can see the stimulus intensity. So here's the stimulus intensity. It stays nice and consistent, which means that the receptor is continually responding to it. And you can see from the fact that you have membrane potentials being generated from beginning to end. This is then how you correspond to the fact that you continuously respond or notice the stimulus. Notice how the receptor potential does decrease slightly, right, towards the end. And that has to do with what we were talking about before, that you can have adaptation to the stimulus. So if we were to increase the intensity of the stimulus, we would see that the receptor wouldn't have a chance to adapt to it. On the other hand, we have a phasic receptor. And as you can see from my phasic receptor, I'm going to go ahead and activate my stimulus. it turns on, it turns off, and what we see happening in this case, instead of having a response that during the entire duration, I only generate a response and a membrane potential at the start of the stimulus. After that, everything kind of disappears. And I won't notice it again unless it goes away and then reappears. So this would be a phasic response because in this case, I only generate an action potential at the beginning of the stimulus. Alrighty, so now that we have established that, let's go ahead and start exploring our different receptors. And I think the chemoreceptors are a nice place to start since we have the majority of our cells that we have in our body can sense some type of chemical stimuli. Within animals, what we see happening is that this would be the perfect opportunity for us to talk about our sensation to smell and taste, so olfaction and gestation. And what we'll do is during the next few slides, we'll take a look how different animal models will have different sense organs. And we're also going to take a look exactly what will control the signal transduction mechanism. So are we going to be using different receptors, different ion channels? Because remember, the goal is for all of our receptor cells to change their membrane potential so that they can communicate that signal over to the nervous system. So let's start with the olfactory system and let's start exploring it in vertebrates. And we'll do vertebrates and insects will be a little bit down the road. Now in vertebrates, what we see happening is that the chemicals that are able to alert the olfactory system are going to be called odorants. And we see that our olfactory system tends to be located in the roof of the nasal cavity. And in order to be completely successful, we're going to need a few things to detect and interpret these odorants. One of them is going to be a mucus layer, and the other one is going to be an odorant binding protein. The odorant binding protein is going to allow the odorants to dissolve in mucus, and once it can do that, it can go ahead and interact with the odorant receptor protein. The odorant receptor protein will thereby be able to communicate the odorant over to the bipolar neurons, allowing for the sensory system to communicate with the nervous system. So let's go ahead and take a look at our picture. So you can see they went ahead and they drew out the nasal cavity of a dog and they've highlighted where the olfactory system is going to be located. And if we take a look at our insert box, we're going to notice that we have our odorant proteins indicated by these little purple dots floating around in a layer of mucus. So anytime you smell something, these odorants are going to be floating around in a mucus layer, and as they do that, they're going to come in contact with our odorant binding proteins, and these odorant binding proteins are going to allow the odorants to dissolve in the mucus, and they will piggyback and bring them over to the odorant receptors. And once they're at the odorant receptors, we see that we have olfactory receptor cells. Those are the sensory cells. Those are our afferent neurons that are part of the sensory system. And they will be the ones that will be detecting the odorant. They will be the ones that will open and close their ion channels. They will have a change in their membrane potential. And they're able to go ahead and communicate it with the interneurons in the olfactory bulb. So that is then its introduction point into the nervous system where it can be interpreted and a course of action can be determined. Also within the olfactory system, we see that some animal models have the ability to detect pheromones. Now, pheromones are often used as chemical signals between animals. They're often utilized to sense a mate, so they're very important during reproductive cycles. They tend to change the behavior of animals, especially if they have to fight for the chance to mate with whoever they feel natural selection is calling them towards. We are not really sure if humans are able to detect pheromones. Some research says yes, others say no, but a lot of research indicates the fact that even if we're able to detect pheromones, because of the fact that we do things like take showers and cover ourselves in lotion and perfumes, sometimes we tend to mask a lot of those pheromones if we indeed secrete them and are able to detect them. So the verdict hasn't been 100% yet on that. But either way, what we can see happening over here in that the mammals that do interpret pheromones, they are going to be using the nasal organ. So it's a different set of tissue and organs that you utilize. They are located at the base of the nasal cavity, not the roof, right? Because the roof is for the olfactory. And then if we take a look at our reptiles, they usually are located within the palate, which is the top part of the mouth section. That is then where they're going to have their vomer nasal organs. And what we see happening is when they're interpreting or interacting with their receptors, they will often tend to rely on a G protein receptor. You guys remember that from our previous chapter? Now the G protein receptor, once it's linked to the proper pheromone, will go ahead and cause the phosphorylation, the activation of the transduction pathway, and then we see the same old story as we saw at the beginning of our presentation. We'll see a change in the opening of the ion channels, which would then cause the membrane potential to be altered, in this case depolarized. And that excitement is then once again communicated with the nervous system. Now, what about insects? How are they able to sense smell? Well, it turns out that they are going to rely on this thing called sensilla. You're going to see this word quite a bit when we talk about insects. Sensilla are hair-like projections of the cuticle. So go ahead and take a look at your picture. You can see the cuticle of our little fly, and what we see happening is we have a bundle of sensory neurons which are able to extend through the cuticle, and this right here is your sensilla. And the sensilla will have a little pore, a little opening on top so that the odorants are able to enter and interact with the sensory neurons. When they make their match with the neurons, they will go ahead and activate the G protein receptors. And what we see happening is that it will activate secondary enzymatic pathways. But beyond that, we're going to see that the story will go back to its original ending, which is the change in ion channels and depolarization. So remember that regardless of whatever the receptor is or whatever the stimulus is that activates it, you will always have the changing of the opening or the closing of the ion channels and the change in membrane potential. I also want to point out that the sensilla, even though you're going to see it quite a bit as we talk about the insects, depending on which system it is responding to, the location will vary. So for instance, when it comes to the olfactory system, we see that the sensilla will tend to go ahead and be located more towards the head section, primarily around the antenna in arthropods. That is kind of where they'll be clustered together, and those will be the ones that will be specific for the olfactory system. Now, how about your sensation to taste? So the gestation system will respond to tastins. Those are the little molecules that they're able to interpret with or respond to. And we have 5 categories. We have our ability to detect salt, sweet, bitter, sour, and umami. And for those of us who've never heard of umami, that's basically what's associated with a meaty flavor, so like a savory flavor. We also know that sweet and salty, as well as the umami, will usually indicate carbohydrates, proteins, and different ions, whereas bitter and sour will usually be helpful in indicating any toxic substances. Now, some of you might have chit-chatted about the sensory system before and talked about the fact that when you look at the tongue, you have what they like to call a little tongue map or a taste map. And basically, it would be an illustration where you have your little tongue, and it would talk about the fact that there are clusters of receptors that would be specific to different tastes. So usually you'll have a cluster of receptors that will allow you to detect salt towards the tip of the tongue. Right behind it is usually when you can detect some sweetness. Sourness is more towards the side, and bitter tends to be towards the back of the tongue. Well, I'm sorry to bust your bubble, but current research has shown that we really don't do range fractioner, or we don't divide up the ranges when it comes to the gestation. A lot of us will not follow the stereotypical clusters of cells. So no, it doesn't enhance the taste of your milk chocolate if you put it towards the tip of the tongue. And no, food doesn't taste more salty if it's all the way at the beginning, or it's more bitter if you shove it towards the back of your tongue, because we see that our receptors to detect our taste will be equally distributed throughout the tongue. At least for many of us, they will. Now, when we look at our taste buds, so for the vertebra, something that I want to point out is that it's actually not going to be a neuron cell. It's in fact going to be an epithelium cell that has been modified. So that's pretty unique. And in fact, we can take a look at our picture right over here. And what we're going to see happening is that embedded within our epithelium cells, we're going to have our taste receptor cell, which, like I said to you before, is a modified epithelium cell, and it will have a pore towards the top, an opening that will allow for the tastins to enter. And once it enters, and the microphylla is there to increase your surface area, once it enters into the taste receptor cells, we could see that at the bottom, it will go ahead and be able to communicate with the afferent neurons, so it can share its changes in membrane potential with the nervous system and communicate the sensation that it has just experienced. Notice also here on the PowerPoint I wrote down for you, each taste receptor can express more than one kind of taste receptor protein. So this is basically to dismiss the theory that only certain sections of the tongue will correspond with certain taste sensations. We can also see that the receptors will tend to be clustered in different parts of the oral cavity, so we can obviously expect them on the tongue, also on the soft palate, which is the roof of your mouth. We also have some by the larynx, and we will have some in our esophagus. Your larynx is your voice box. That's the vibrating part of your throat. On the side right here, I've included a picture of a little fish, and that's because it's actually quite common that you can have some taste buds that will be located on the barbels, so these little extension whiskers, as well as on the fins. So as the fish is engaging with its environment, they're able to detect tastings from different locations. In invertebrates, they will not be using specialized epithelium cell. They will rely on good old-fashioned sensory neurons. And also what we see happening with the invertebrates is, once again, we have that projection that's going to come from the cuticle, the sensilla that will be utilized. Now, in this case, we see that for taste, you're going to have a lot of them that will be specialized around the mouth region, but also around the wing and the end of the legs. And I want to point that part out because have you ever seen where you have maybe you had food out. Maybe we were doing a picnic. Wouldn't that be nice? We'll have an outside picnic. Maybe we have a fruit salad on display. If a fly lands on the food, I don't know if you guys have ever noticed how sometimes they'll scratch their legs together, or am I the only one looking at the flies instead of shooing them away? But either way, the next time you see a fly, observe its interaction, and when you see that it's scratching its leg as it's sitting on the food, that's actually one of the ways that it can taste the molecules that are in the food and enjoy the sensation of taste. Isn't that kind of cool? Now, when these taste buds are activated, keep in mind that with the invertebrate, they are neurons, not modified epithelium cells like what we have. And when the sensory neurons are utilized, remember the goal is once again to cause a change in the ion channels and the membrane potential. Also, a little statement at the bottom, because there are such a wide degree of differences in taste sensation between vertebra and invertebra, it has been stipulated that it's most likely due to independent evolution. Alrighty, let's now do mechanoreceptors. So mechanoreceptors are going to be the ability for us to notice things like pressure changes, touch, vibrations, positions within the body. The main goal of a mechanoreceptor is to take a mechanical stimuli and convert it into our electrical signals. And the electrical signal is the fact that we're doing our depolarization and we're generating an action potential. In order to do this transformation, we see that the mechanoreceptors usually come in two types. You can have what we call an epithelium sodium channel based mechanoreceptor or a TRP. The TRP stands for transient receptor potential channels. Now, regardless of which one you have, so the one that will be responding to sodium changes, here's your little sodium channel, or the one that will be responding to the transient receptor channel, we're going to notice that all of them will have a linker to the cytoskeleton. And the cytoskeleton obviously is what's inside of the actual receptor cell within its cytoplasmic content. And this, of course, is going to be extremely essential because remember, we're trying to radiate the signal from the outside to the inside of the cell. So all of the channels will be linked both extracellular and intracellular. Extracellular is how we're going to be responding to our stimuli, and intracellular is how we're going to be propagating the ion changes and the change in membrane potential. Three different classes when it comes to our mechanoreceptors. We're going to have our baroreceptors, our tactile receptors, and the proprioceptors. Your baroreceptors are all about detecting pressure changes. So your body will, for instance, utilize that within the walls of your blood vessels to see if you have an abnormally high or low blood pressure change. Your heart will utilize it to detect how much pressure is being placed on it as it's going through its systolic and diastolic motion, contracting and relaxing. Your digestive system and your urinary system will often use baroreceptors to maintain a flow of items. So for instance, for your digestive system, it's able to detect when it's time to release chyme, partially dissolved food, or bolus from the stomach, chyme from the stomach into the duodenum, the top part of the small intestines. The urinary system can utilize it to figure out when it's time to open the sphincters so that urine can flow out through the urethra. Tactile receptors, on the other hand, those tend to be more towards the body surface. So they're the ones that we can utilize for touch, pressure, vibration. And proprioceptors, as I was mentioning to you before, is the ability to kind of figure out where your body belongs in space. So it's a fantastic way to monitor the position of the body into relationship to itself, as well as the environment that you're currently in. In fact, let's go ahead and take a few seconds and learn a little bit more about our tactile receptors. These tend to have a lot of isolated sensory cells that will be embedded in the skin. And in fact, some of you might have seen some of them before. So have you guys ever learned about the Merkel discs that are associated with the Merkel cells that you have in your epidermis? These are the ones that are able to communicate with the nervous system through neurons. You can see them right over here in the dermis, the middle layer of the skin. And what we see happening with the Merkel cells is that it's a relatively small receptive field, so it's excellent at discriminating very small amounts of stimulus. And in fact, when someone learns how to read Braille, they are able to sense the stimulus because of these particular Merkel discs. There are other receptors that we have hidden within our skin. One of them is, for instance, the Ruffini corpuscles. The Ruffini corpuscles are excellent in allowing to communicate the location of the body in space. So exactly how far your body is from a certain piece of furniture. The reason I bring this up is because I was just thinking about the fact, if you're anything like me, you probably set your alarm with your cell phone. And for me, I'll set my alarm with my cell phone Who am I kidding? I set 10 alarms on my cell phones because it's so hard to get up in the morning for me. But either way, I set my alarm on the cell phone, and sometimes I'll catch myself that I'll wake up and I've completely missed all the alarms, and it turns out that I hit snooze every single time, but I don't remember it. So how does my body know exactly where my phone is going to be located? That is all due to the funny corpuscles. It keeps track of space and where your body is located within it. So the best thing to do if you find yourself ignoring your cell phone alarm, or you find yourself hitting the snooze button and falling back asleep even before you realize it, is go ahead and move the location of your phone on a regular basis, making it harder for your raffinate corpuscles to keep track of it. The other one, the perennial corpuscles, the ones that are on the side right over here, those are excellent at detecting vibrations. So if you have your cell phone in your back pocket, for instance, or even if you're holding it in your hand, you might notice that you find yourself reacting before it actually fully vibrates, and that's because of the perennial corpuscles that are able to give us a heads up that the phone is going to start doing that particular motion. When it comes to the insects, I don't think it will be a surprise to anybody that we're going to be utilizing our sensilla. There you go. Now, in this case, when it comes to the sensation of touch and pressure, we do see that they come in two variations. You can have the hair-like projection, which we're used to seeing, and we're going to refer to that as a trichoid. And the trichoid will be able to detect the stimulus and communicate it directly to the bipolar sensory neuron. As you can see it right over here. And this will then go ahead and activate a transient channel, a TRP channel. On the bottom one, you see that we can also have a domed shape projection within the cuticle. And in that case, we're going to call that a camniform. And this one will also communicate with a bipolar sensory neuron. To be very honest with you, both of them will have the same reaction, meaning the bipolar and the channel activation. But really what the difference is with the receptor is whether it's going to be a hair-like or the dome shape. So really it's the name, the trichoid, and the camniform that are the most important things to highlight when it comes to the insect tactile receptors. We can also see that the proprioceptors in the insects are going to be modified. They're going to be different than the traditional tactile receptors. And part of that is that it's going to be part of the structure called scoloppedia. Scoloppedia takes a look at the fact that it's able to detect any type of bending within the cuticles, and it does so by interacting and attaching to the cuticle with the use of a cap cell or a little ligament cell. And usually within the stack of items, we're also going to notice that they have bipolar neurons that are able to detect the sensation, and also we can see that it can be modified to turn into a tympanial organ. The tympanial organ will then acquire a different function. Instead of only focusing on proprioceptors, it will also be used to amplify sound detection. So we definitely go ahead and we'll explore that a little bit more. But for now, if you see the term scoloppedia, I want you to think about the fact that that is what insects use for their proprioceptors. Let's go ahead and stop right here for part one. And as I said to you before, make sure that you are taking your notes. And if you have any questions, definitely don't be shy, reach out. But I will hopefully see you guys in a little bit. Let's go ahead and we'll continue on with part number 2 of chapter number 6 whenever you're ready. Bye, guys.